folks on the phone. Welcome to the Department of Medicine Town Hall. We're going to tonight talk about the use of monoclonal antibodies. A lot of patients and a lot of faculty have approached me and asked me questions about monoclonals, that some of which I've been able to answer and some of which I've deferred to this town hall. So Adit Ginde is a professor of emergency medicine in the School of Medicine here at the University of Colorado. And he has quite a bit of expertise on monoclonal antibodies and is involved in a number of therapeutic trials. So Adit, you're, I assume, going to present for about 20 minutes or so, and then we'll open it up to discussion.
Yeah, sounds great. Thanks, David. Thanks, everyone, for joining this evening. Hopefully you have a glass of wine or a beverage of choice with you. And as David said, we'll go over next 20 minutes or so an update on monoclonal antibody treatment for COVID-19. I last had the opportunity to present in this forum about 7 months ago, and I'm going to sort of pick up from there and catch everyone up to speed, and then get to the current state. There's a lot that's happened in this space during that time, and then hopefully we'll have a really nice opportunity for discussion and probably as many questions as we have answers, but that's what these forums are really good for, I think. So just some disclosures quickly. So we have a couple of NIH grants for monoclonal antibody work. There's an NCATS supplement to fund the MAB Colorado project, which we'll talk a little bit more about during this presentation, essentially for dissemination, implementation, and real world effectiveness work. And then I've been involved in the Active3 platform, started with Operation Warp Speed and then transitioned since last June, which is looking at monoclonal antibodies in the inpatient setting. Other funding is listed and there's no conflicts of interest to declare. So I think we're all familiar, but just so we're on the same page as of today, at least, where we are in terms of the pandemic. So the red line is cases and the orange line is hospitalization. So we know we've been in the thick of the Delta surge over the past couple of months, starting to feel like it's coming down, at least in the US. Seeing some cases come down in Colorado, although hospitalizations have plateaued but not started coming down. These is daily hospitalization rate. We have seen hospitalizations start to come down at UC Health. So every time we have one of these downturns, there's a feeling or a hope that we won't have a need for therapeutics in the same way, and then another surge occurs. And that certainly happened during the early summer, where there was some hope that we would have a sustained plateau, and it just didn't happen. And I don't think now anybody has much faith that this winter will remain plateaued, but we can enjoy our downturn in the coming couple months a little bit. Part of the issue here is vaccinations in the US and in Colorado. And this daily vaccination rate, it's hard to see the axis, but just look at the trends that we're certainly plateauing and starting to come down in terms of daily vaccinations, and do not have enough of the population vaccinated at this point, as we know. So here's a spectrum of illness of COVID-19. We're going to focus most of the discussion on this mild symptomatic illness, so outpatients, where antivirals is really at the heart of treatment. And the goal primarily is to prevent hospitalization, but also to enhance recovery. And we'll look at a little bit of data that looks at symptom recovery as well. But we'll also start to, and this is some of the new data from the last several months, look at the asymptomatic population, or look at the even the exposed population for prophylaxis, and then even the hospitalized patient population, which we thought was previously off the table as it came to monoclonals, and there's some emerging data that I think will inform our practice. So to get everybody on the same page, neutralizing monoclonal antibodies for COVID-19. So I'll abbreviate it as MAB, but the neutralizing is important because there's other monoclonal antibodies that are not SARS-CoV-2 directed. We're specifically talking about SARS-CoV-2 neutralizing monoclonal antibodies. From the very beginning, it was a focus of federal efforts, Operation Warp Speed, robust public-private partnerships in the therapeutic domain. These are laboratory-made, isolated from recovered patients, neutralizing IgG antibodies that are directed at the SARS-CoV-2 virus. The ones available all bind to various parts of the viral spike protein to generally prevent interaction with the ACE2 receptor, which is the primary way for cell entry. Schematic over here. Several agents are combination treatments or cocktails interfacing with different aspects of the spike protein. The idea is synergistically they can increase treatment efficacy, but also importantly prevent escape mutations. And the three available agents are casirivimab, imdevimab, which is Regeneron's, bamlanivimab, etesivimab, which is Lilly, and sotrovimab, which is the Vir molecule that's marketed by GlaxoSmithKline. I'll refer to them by company, not because I have any affiliation, but because it's quicker to say in the rest of the talk. So I'm going to remind where we were the last time we spoke about monoclonal antibodies, which was early 2021. Here are the data that we were working with. So it was a New England Journal paper in late October that was phase 2 data for the Lilly monoclonal. Primary endpoint was viral load, actually at 11 days, and there was no difference. There was some difference at 3 days. And then there was a focus, and this really started to pivot attention towards prevention of hospitalization. There was a reduction. This was dose finding, so there were 3 doses, but essentially 6% versus 1.6% hospitalization rate. The challenge was numbers were small. We're looking at 9 versus 5 hospitalizations with a 2-to-1 ratio of patients enrolled to the active treatment. So it was still quite preliminary, but led to an emergency use authorization by the FDA in November for use of bamlanivimab monotherapy for high-risk patients with COVID-19. This is based on a few hundred patient phase 2 trial, but sort of grasping for treatments that might have effectiveness. Soon thereafter, now by press release in late October, New England Journal article in December, there was a Regeneron dual combination monoclonal that had now 800 patients. There was a high dose, low dose, and placebo. This was interim results from the phase 2 components, which showed a 57% reduction in COVID-related medical visits, and a particular or potential increased signal for higher risk patients, 72% reduction. And this led to their emergency use authorization a few weeks after the Lilly agent. But we were, in a different situation. So back earlier this year, there was concerns about limited availability of drug. They were still needing to ramp up manufacturing. So actually, the state of Colorado, in partnership with some experts from University of Colorado, including Matt Winia from the Center for Bioethics, developed a random allocation system. Never actually really had to trigger it, and that was because there was really low uptake of the treatment. Far less than 5% of eligible patients were accessing it. There were some equity issues. And I use the past tense, but all of these sort of are still issues even today, where mostly non-Hispanic white patients were accessing treatment. Only really those that had robust access to care or even personal connections were getting treated. The one area that we've addressed, I think, now in the last 7 months is at that time, there was still quite a bit of uncertainty about benefit and even risk. We were dealing with relatively small phase 2 trials. And so that was creating some hesitation, and then, vaccines were taking hold, and it was sort of pushed off to the side a little bit from a therapeutic standpoint. So we're going to march forward. So this is new data from the last time I presented. So this is the phase 3 data from the Lilly trial, now combination agent of BAM and EDI, which now shows an 87% reduction in hospitalizations by press release, hospitalization and death. We had a Regeneron press release showing a 70% reduction, this time March 23rd, 2 weeks later. You see the percentages of hospitalizations. This is the first time we're seeing symptoms. So this whole recovery concept, a 4-day median reduction in symptom duration, really important for patients, especially because relatively few actually ultimately get hospitalized. And then around the same time, the VR molecule marketed by GSK showed in the Comet Ice phase 3 trial 85% reduction in hospitalization or death. Really uncomfortable to go off of press releases when we're trying to make clinical decisions, but that's sort of the era that we've been in. And actually led, if you look, these are all in March, April 8th, the NIH treatment guidelines upgraded their recommendation for the 2 monoclonal agents that had emergency use authorization to actually class A evidence, where it remains today, for outpatients to prevent clinical progression to hospitalization or severe disease. Still to this day, there's only one other intervention from a therapeutic standpoint that is a class A intervention, and that's corticosteroids, or in particular dexamethasone for critical COVID or those with high flow oxygen or mechanical ventilation. So there's actually very few that have this level of evidence, and but still there was a lot of confusion going on here, and I'm kind of walking through the journey. I will get, I promise, to a distilled message at the end of the current state. But I think it's important because I think there's been a lot of confusion about what the state of evidence has been over this year. So here was a release from the FDA. This is now 1 week after the NIH guidelines enhanced their recommendation to Class A, and said the big headline, revoking the EUA for bamlanivimab, and everybody was worried that what's going on. If you look at the fine print, it's actually just looking at monotherapy because of emerging resistance from the alpha variant at that time. And there was still endorsement of the Regeneron combination agent and the Lilly combination agent as well. But this kind of this type of release with this bold headline, when people are not, we're trying to absorb as much information quickly, was a little detrimental to progress. Now we're starting to see some more robust data. So here's the phase 3 trial results for Regeneron, which came out in a preprint release on the medRxiv platform. Still has not come out in a peer-reviewed journal, although is expected within the next month. So this is 4,000 outpatients with at least one risk factor for severe disease, tested 2 doses, 1 of 2 doses versus placebo, followed patients for 28 days. And here are the results were a 71% reduction in hospitalization. Again, vast majority don't get hospitalized, but still a remarkable result in a 4-day reduction in symptom resolution. It's very important as well. Well tolerated in these 4,000 patients, and actually the DSMB stopped the trial for efficacy. Here's what the data looks like in a Kaplan-Meier time to event curve, where essentially can see that most of the hospitalizations are occurring in the first several days, especially with the active agent, still occurring in the first 2 weeks, but really nice separation between the groups. And here's the time to symptom resolution, where you see an enhanced time to resolution with no real difference between the 2 doses that were tested. Because the lower dose looked about as effective at the 1,200 milligram dose as the 2,400 milligram dose, the FDA then authorized the lower dose for use for treatment of outpatients, high-risk outpatients with COVID-19. And then also in this release, this is now we're getting into June, the subcutaneous dose was now authorized as well. So you can give the same formulation subcutaneously. It's 4 injections at different sites, so it's not the easiest, but infusion capacity giving intravenous treatment can sometimes be limiting. So that was helpful for some settings where IV access was not easy. And this was based primarily on virology data, not on clinical outcomes in the subcutaneous formulation. And now we're at the end of June. Now we're pausing the distribution of BAM-EDI. So this was still authorized, but Asper, which was delivering monoclonals by direct order at that time, basically put out this release that the combination Lilly agent was, there was increased resistance to the gamma and the beta variants, which were increasing in prevalence in the United States at that time. So then it was sort of a Regeneron-centric approach. Sotrovimab, the GSK agent, was available, but there was not a federal purchase and still is not. Whereas the Regeneron Lilly agents had a large federal purchase. So the drug is free for those. So really this became sort of all Regeneron at that point in late June. A couple weeks later, the Lilly combination agents, there was a publication in the New England Journal of their phase 3 data, which is 1,000 patients, 1-to-1 active versus placebo. Pretty similar results, 70% reduction in hospitalization and death, no difference in adverse events between the groups. Here are the time to event curves. You'll see that these just look very similar across agents, and shows really nice separation between group for prevention of hospitalization and death. Now we're getting close. We're getting into the last month. So now early September, you could see that's kind of a roller coaster story. Now we're resuming the distribution of BAM-EDI. So it had been sort of on pause for 2.5 months. That was really because with the Delta variant, for most mutations in the Delta variant or the prevalent variants, there was effectiveness and no evidence of resistance to the agent. So it was redistributed. And then today, there was a reminder that we're seeing an increase in the mutations in the Delta variant that cause resistance to BAM-EDI. And so to be very careful, and you have to go state by state to know whether this is available to you. In Colorado, this is available, and the prevalence of those mutations are quite low at this time. But again, really hard to sort all of this out. So that's where we are. I'll kind of summarize that in guidelines in a few minutes for outpatient treatments. There are some data on post-exposure prophylaxis as well. It's a JAMA paper published in early June with the bamlanivimab monotherapy. This is in skilled nursing facilities and assisted living facilities, either for residents or staff, where there was at least one positive infection at that facility. 1-to-1 placebo or IV BAM, followed patients for a couple months and saw a 57% reduction in symptomatic SARS-CoV-2 infection, with maybe a greater effect in the residents of the facility versus the staff of the facility. I think, here it's still low numbers, but you can see a really nice separation between groups in terms of the prevalence of new infection. And not a lot of separation, at least start to see a little bit of separation here amongst the staff participants of the trial, but lowish numbers, so hard to compare between the two. Similarly, early August, this is the prophylaxis study for Regeneron's agent. This was all subcutaneous dosing, 1-to-1 versus placebo. This was now household contacts, so not facility contacts, but basically a member of the household had a symptomatic SARS-CoV-2 infection and enrolled patients that were within the household, and an 81% reduction in symptomatic infection amongst those household contacts that were not yet infected, and nice separation between the groups here. Not a huge, kind of a surprisingly low percentage of the placebo group got a symptomatic SARS-CoV-2 infection, but these were also clinical trial subjects that I'm sure were quite careful with household exposures. And then Regeneron's data goes nicely. They did a nice job of collecting symptoms in all of their trials. So here is if you did get infection, and there's only 11 infections in the treatment group, 59 in the placebo group. The duration of symptoms were much less as well. And you'll see that theme that even if you do get an infection, your symptom duration is lower if you do it in the treatment phase, in the early treatment with outpatients, they get better quicker. They are less likely to progress to severe disease. So, and then the FDA expanded the emergency use authorizations for both of these agents, for Regeneron and the Lilly, for prophylaxis. Regeneron is subcutaneous. Lilly's agent is still intravenous, which causes some logistical challenges. And then to round it out, what about hospitalized patients? We felt like this is off the table for a long time. Here's our trial within the ACTIV-3 platform, looking at bamlanivimab monotherapy. That we looked at recovery rate ratio, so that the primary endpoint was time to sustained recovery, defined as being discharged from the hospital and going home to your prior location, staying home for 2 weeks, so preventing bounce backs. And essentially no daylight between the treatment and the placebo group, with about as null a result as you can get overall. And these were the operating data that we were going under. Maybe this just doesn't work in inpatients. We published some more data in preprint server that is should be coming out in peer review very soon, which is starting to get at a new concept here, which is that baseline serostatus, or your antibody status, at the time of treatment in hospitalized patients can actually be predictive of treatment response. So higher is better. So we're talking about recovery. So if you're antibody negative upon study entry, you're starting to see perhaps a separation between the groups in favor of benefit. Again, smallish numbers, only 350 patient trial. We were stopped early for futility. Confidence interval across is 1, but you'll see some more data to support the story. And then a harm signal potentially for the antibody positive patients at baseline, where there's slower recovery. Again, don't want to overinterpret, but there is a p-value 0.02 for the interaction between baseline serostatus. Here are the data a different way. A secondary endpoint of a composite of deaths, serious adverse events, organ failure, co-infection. Not too much going on between in the aggregate group, but now we're looking at antibody negative. For this outcome, higher is worse because these are bad outcomes, and actually seeing potentially lower event rate in the treated group if you're antibody negative, and a higher event rate in the treatment group if you're antibody positive at baseline, with a significant p-value for interaction as well. Again, don't want to overinterpret single trial with modest sample size, but this is starting to tell a little bit of a story. And then in June, inpatient data from the recovery platform in the UK that tested the Regeneron agent came out. So pretty similarly, so this is again still on preprint, not out in the peer review literature yet. Similar story in overall. So this is all participants, and they, it's a pragmatic trial with relatively limited data collection and open label. So there's some methodologic differences. A larger sample size, not much difference between treatment and control. But then you look at seronegative versus seropositive. So the top 2 lines are seronegative. So the first thing you'll notice is if you're seronegative in the hospital, it is a bad prognostic sign. So if you've not yet mounted an antibody response but are sick enough to be hospitalized, your mortality, and this is mortality as a primary endpoint, is way higher regardless of treatment than if you're seropositive. But if you're seronegative, you have a markedly lower mortality, pretty hard endpoint, inpatient mortality than their control group usual care. And again, maybe a suggestion of a harm signal, not statistically significant, but for treating seropositive patients with a monoclonal antibody. Regeneron has actually filed for an EUA for inpatients in seronegative patients, which is anticipated pretty soon because these data are fairly compelling. So we might start seeing a change in practice soon in inpatients. One of the challenges is going to be which antibody assay should we use and how quickly can we get it? Because I think there's pretty compelling evidence from these and other trials that seropositive patients are unlikely to benefit and actually concerned that they may be harmed. So would not want to give this treatment to seropositive patients. And then patients enrolled in both of these trials, about half of the patients were seronegative at trial entry. And probably it would be even more than that at hospital arrival, since they're enrolled a day or two into their hospitalization. Why might this be happening? There's this concept of antibody-dependent enhancement of disease, particularly in viral infections. I'm sure there's others on this call that are quite expert in the immunology of this. But essentially, if you have the two primary pathways for this, where essentially paradoxically antibodies can make the disease worse. One is a viral replication paradigm, which is you have a non-neutralizing antibody that, because of its Fc effector function, can actually facilitate entry into the cells and enhance viral replication, despite the intention of these being neutralizing antibodies. We are actually currently testing an active 3 in a monoclonal made by AstraZeneca that has a modification that avoids Fc receptor interaction. So that will be some really nice data soon, actually, to look at this particular question empirically. And then the other is sort of an immunopathogenesis argument, which is formation of immune complexes and potential for essentially a exacerbated inflammatory response through multiple mechanisms that could be potentially quite relevant in the airways. Again, all theoretical and quite difficult to measure. Okay, so we're going to round it out and open for discussion. So NIH treatment guidelines, where are we right now? But updated September 24th. If I gave this presentation in a couple of weeks, this may be different, but this is what we have right now, which is it's been updated now for all 3 monoclonals that have a EUA, recognizing that GSK's agent, sotrovimab, does not have a federal purchase. So you actually have to purchase it directly from the supplier. That may change in the next couple of weeks. But essentially, strong recommendation for non-hospitalized patients that do not require new supplemental oxygen in the outpatient setting, with a particular focus on high-risk patients. And this update was provided a few weeks ago by the panel, which was recognize that there's either shortages of the drug, which has been happening some, shortages of supply chain, which has happened in the last 2 weeks since the federal government shifted distribution to the state level, didn't necessarily warn anyone. And the other is just logistics. It's hard to have enough staff to administer this agent on a mass level, especially intravenously. So there's some suggestion of how to prioritize. One is to prioritize treatment over post-exposure prophylaxis, just to do the most potential for good. The second, which has created a lot of discussion, I'm sure we'll create discussion today, is to prefer either unvaccinated or incompletely vaccinated individuals or vaccinated individuals who are immunocompromised over other vaccinated patients for treatment. This creates a little bit of moral distress among some providers because, and patients as well, due to, some patients will use this, and even some states and their governors will use this as a replacement for vaccination. Also, those that quote did the right thing and got vaccinated still get sick and are still at risk for severe disease, may be denied treatment in favor of unvaccinated individuals. So I think, a tricky ground, but the guideline committee is really taking a most good for the most people in a limited situation approach. Bottom line, monoclonals are available, can be somewhat challenging to get for your patients, efficacious for symptomatic positive patients, not hospitalized, not on new oxygen therapy or increase in baseline oxygen. Ideally, really giving within the first 7 days of illness, although it's authorized up to 10 days, and have at least one risk factor for progression to severe disease. Won't show you all the EUA criteria, but essentially it's any comorbidity, elevated BMI, and a high-risk group, including racial and ethnic groups or healthcare workers. So there's actually about 80% of people that are outpatient positives will be eligible for treatment. It's actually hard not to be eligible for treatment. Prophylaxis is potentially available, although there's a preference for treatment. And then I suspect coming soon is treatment in seronegative hospitalized patients. Give a couple of quick words before we close on our MAB Colorado project. This is the NCATS project, where we're doing a hybrid implementation effectiveness trial, really looking at, we're taking an understanding that we know about efficacy in a clinical trial setting, but a paradigm where we're having trouble giving this to patients in a mass scale. So assessing barriers and facilitators to use statewide, looking at strategies to implement, especially with equity, this treatment, and then simultaneously looking at real world effectiveness data. We don't know anything in patients about how this works in the Delta era, and we have some really compelling data coming soon to inform that question. Move forward, because I think we're over time. We have a nice website that has some really great resources for the community and providers here. Some sheets for providers to kind of help, flow sheets, give some reminders about risk conditions. For those, we'll talk a moment about implementation in case this comes up, we can come back to it. There's over 30 infusion sites throughout the state of Colorado, including 4 within the UC Health system, with more coming quickly, or more being expanded in the next couple weeks. But we also know it's very difficult to get this for patients. This was a provider survey that was led by our D&I core lead, Bethany Kwan, that really sort of outlined the challenges that providers have in ordering treatment, how long it takes, concerns about lack of clarity on out-of-pocket costs for the infusion itself, not enough knowledge, and potential for getting to patients too late. So the thoughts are really to simplify and centralize the ordering process, and just disseminating more information, which our team has been working tirelessly to do. We've really partnered nicely with the Virtual Health Center at UC Health for the centralized strategy. So for providers here, you can order this yourself, and there's been several blasts from UC Health leadership about how to do that. You can also refer patients to the Virtual Health Center, who can, across the system, get access to treatment for patients. Because we're still limited on infusion slots, there's still a preference for older adults over age 65, those with 3 or more eligible comorbidities, immunocompromised patients, or now an increased focus on unvaccinated patients as well. Denver Health has been a great partner in this work as well. Lindsay Fish, who's the director of the Pain Urgent Care, where Denver Health is doing their infusions, been very involved partnering with Denver Public Health as well. And then Tri-County Health, we've been partnering with to refer patients in our surrounding counties that are not necessarily UC Health patients, and some other activities related to creating a patient-facing call center, engaging urgent care centers and home health providers as well. So sort of a multi-tiered approach to implementation. And if you didn't see yesterday, Tri-County, actually CDPHE, in collaboration with our MAB Colorado team, put out a HAN advisory, and health advisory alert, that was essentially all about monoclonal antibody treatment. They did a really nice job of piecing together the current information. So recognizing that 2 weeks ago, there's a decentralized approach, so now ordering through the state has to occur again. There's some information about how to refer patients for monoclonal antibody therapy. There's some more tailored information at the health system level as well. And then if you want to procure a drug, they have information on the ordering process as well. Here's our MAB Colorado team. It's a big team. We've been working together for 6 months. A lot of people in the Department of Medicine has contributed greatly to this effort, and just a lot of, I think, really important activity in the dissemination, implementation, and clinical spaces as well. All right.
Good. Adit, that was really great. Lots of material, lots of new information and recent information. The one area that you didn't bring up is prophylaxis in individuals who are immunocompromised and have not responded to vaccines. Tell us, if you can, just summarize that, and then we'll get to the other questions in the chat box.
Yeah, so treatment of asymptomatic, non-exposed patients is still a little bit controversial. Theoretically, there's evidence in this, particularly from the new AstraZeneca monoclonal, that a long-acting monoclonal can be used essentially as a vaccine substitute or a vaccine augmentation. But this isn't really sort of an authorized approach yet. The AstraZeneca monoclonal has a half-life of 2 to 3 months. There's a mutation in the monoclonal structure that extends the half-life. And it does work, and it does prevent infection for an extended period of time. Wasn't tested specifically in immunocompromised patients, but would be a potentially nice option. It's not covered under the emergency use authorization. So for individual patients, it would have to be sort of a expanded access, one-off compassionate use type of scenario, which is a little bit difficult in a prophylaxis scenario. But I do think that there's forthcoming work in this area, because I think we know that immunocompromised patients are struggling to create or maintain a adequate immune response, at least as a large group, still having some protection, but we're still seeing a number of hospitalized breakthrough infections as well. So I think more to come, not quite ready really for prime time. So that's a short answer.
Yeah, I think, it's a quality of life issue for immunocompromised individuals that have not responded to the vaccine as well. So,
yeah,
okay.
No, in terms of what they feel safe doing, and
exactly. Yeah,
absolutely.
Okay, Steve Johnson, you have a question for Adit. Why don't you go ahead?
Yeah, first of all, thanks, Adit. And I'll mention, because I don't think Tom Campbell's on, he does have an upcoming protocol looking at the Regeneron cocktail as a prophylaxis among immunocompromised folks, I think vaccine non-responders. My question is that, as time goes by, more and more people have been vaccinated, and the subgroup that have not been vaccinated will have infection, and vaccinated people will have breakthrough infections. And so I'm wondering what we think about the performance of these monoclonal antibodies in a world where a lot of people have antibody either induced by vaccine or induced by infection. And do we need to repeat some of these studies now in groups that are highly exposed through vaccine or infection, as opposed to using studies that were done really prior to this era?
It's a fantastic question. I spent the last 2 days in a NIH summit on the future trials of monoclonal antibodies, and this was about a third of the discussion. You're right, the existing trial data were an era where less than 5% of the trial participants were vaccinated. Our AstraZeneca trial that actually ends enrollment tomorrow will be about 25% vaccinated patients. So we'll get a little bit of a sense. This is in hospitalized patients. But the idea is to design the next level of monoclonal trials or other passive immunity or antiviral trials in the context of stratification by vaccination status, and with a particular focus on oversampling immunocompromised patients. Not only, as you said, for the upcoming work for prophylaxis, but even in the treatment paradigm. It's going to be challenging because it's hard to withhold treatment from patients. So right now, we have the EUA in outpatients. I suspect the EUA is coming soon for seronegative inpatients. So it starts to get a little bit challenging to do clinical trials where you have an overlapping treatment mechanism with the treatment thought to be effective. So I think that's going to be a challenge. But I do think that it's really hard to know how these treatments that were tested in a post-vaccination era work and respond to, pre-vaccination era in a post-vaccinated population, recognizing that there will still be unvaccinated patients. Sorry, that was a long-winded answer to say that's a great question to study.
And that it's something that really needs to be studied.
Absolutely. It is planned to be a focus going forward for sure.
Good. John Gutman, you have a question.
Yeah, it was maybe a little long to type. I'm curious about maybe a little more nuance around hospitalized patients, because I mean, my sense, the data on these things is really compelling, I think, given early. And I also feel like my sense is that, you know, this is a really time is of the essence situation to get the benefit before that inflammatory response gets totally out of control, and there probably is no benefit. So when it comes to the in-hospital data, I can totally buy once you're in the ICU, there's no value, but how robust do we have a sense of sort of, you know, that first 24 hours maybe, and then a follow-up on that too, because I deal with the severely immunocompromised. Do we have any data on seronegative because you have not been exposed to COVID versus the immunocompromised seronegative patients when they're in hospital? Because we've had some patients who are in hospital who get it, and we kind of can get into some debates about whether we can give it to them or not. They've been pretty liberal, but it can be challenging.
Yeah, they're really, really good questions. We went into, it's hard to go into clinical trials thinking that the results are going to be positive, but we were pretty surprised about the results in aggregate in hospitalized patients. And I don't think it's because we were getting patients too late. The median duration of symptoms was 7 to 8 days. The median time from hospitalization to enrollment was 1 day. So, it takes a little longer to enroll a patient than perhaps to give a clinical treatment, but it was not that bad. So I think I think my read on the current state of the data are that if you have a bunch of seropositive patients in your cohort and feel very convinced that seropositive patients as a group are unlikely to benefit, that's hard to see an aggregate signal when you have half of your patients with antibody. And those generally are not because of prior infection or vaccine-mediated immunity. It's largely because they've progressed enough in their disease to mount their own host immune response to that given infection, which obviously is more challenging in immunocompromised patients, which is why it's a group of interest. There's emerging data that characterizes the prevalence of seronegativity and positivity in immunocompromised patients in our trials. And it's certainly much higher. But, getting down to a seropositive immunocompromised patient, are they unlikely to benefit or potentially being harmed? Getting into pretty small subgroups, so we have to borrow from the bigger cohort. I do think that it's not unreasonable to think that this therapy is going to be given very soon, but I think that we're going to need an antibody test. The challenge is there's a number of antibody tests, and they measure different things. Some are functional, some are nucleocapsid, some are anti-spike. Which one do you use? They sort of give different results. I think makes it challenging, and which ones are going to be available at the point of care or in rapid fashion to make a treatment decision. So as you said, ideally this treatment, just like any other source control when there's pathogen replication, should be given as soon as possible. So sitting around waiting for an antibody test result for 24, 36 hours doesn't feel great either. But I think you're right. I mean, there's a feeling that early in hospitalization is the right time prior to progression to critical illness. You're still sort of in the viral replication phase. We do see much lower viral loads and antigenemia in the seropositive patients. So I think the thought is that they're already well on their way to clearing the virus through their natural host immunity. And so probably adding monoclonal antibodies on top of that is not going to be particularly beneficial.
So Susan Boakel has a similar type of question focused on the antibody responses. And her question is, if the individuals who are hospitalized with antibodies do worse, what about, how should we think about the outpatients with and without antibodies who have been vaccinated?
Yeah, it's a really good question. I think we're going to have some data from our real world effectiveness study very soon. We actually just got a data transfer on 6,000 patients from the Colorado Vaccine Registry. And we'll be looking very carefully at vaccinated versus unvaccinated patients who did and did not get monoclonal treatment. Totally open question. I will say that the only data that I saw in the last couple of days to inform this question came from the Regeneron outpatient trials, which essentially serostatus in the outpatients did not seem to be particularly predictive of treatment response like it did in the inpatients. So perhaps it's still early enough before they progress to severe disease that it could still be beneficial and hopefully not harmful. But I don't think we have a clear biological explanation. There's a lot of theories and hypotheses floating out there, but for why the seropositive patients may actually be harmed by the treatment in the inpatient setting. So I think then extending it to vaccinated patients that haven't really been robustly studied with this treatment, I think becomes sort of an open question. And perhaps is one of the reasons why, maybe Steve Johnson can comment if he's allowed to, but why the guidelines committee, one of the reasons why there was a focus on unvaccinated and immunocompromised patients for the treatment. I just don't think we have enough data to inform outpatients that are vaccinated. And certainly we've been referring them for treatment if they can get it and they're high risk.
What's the current recommendation about monoclonals and how soon you can be vaccinated after you receive monoclonals?
So the current recommendation is still waiting 3 months after receiving a monoclonal before getting vaccinated. There's still really not adequate data or really any data to inform this question. It's really based on the theoretical potential for a reduced response to the vaccine if you get vaccinated too soon after a monoclonal antibody treatment, but not informed by data. And it's in combination with the fact that if you get monoclonals and have, or really have natural immunity from an infection, the chance of reinfection in the first 3 months and really in the first 5 months after that event is exceedingly low, such that it's worth it to wait, make sure you get a robust immune response to the vaccine. But again, I think this can be tailored. There are certainly specific patients, immunocompromised patients or others that there may be a desire to vaccinate early. But that's still the current recommendation is 3 months.
Let me just get back to my initial question about using monoclonals as a prophylaxis in immunocompromised individuals that have not responded to the vaccine. If that were to come to pass, how frequently would an immunocompromised individual need to receive an infusion of monoclonals?
So it depends on the monoclonal. So Regeneron and Lilly's monoclonal, the half-life is 2 to 3 weeks. So it's really not a great agent for this purpose as sort of vaccine replacement, because if you take a few half-lives, you're infusing this every couple months. But there's the AstraZeneca monoclonal in particular, which has actually been tested in this paradigm, and the half-life of around close to 3 months. It could be more in the 6 to 9 month range, which is maybe where we're at from a vaccination of other patients standpoint in terms of needing boosters. So it gets into a more realistic time frame.
And you didn't mention side effects. Tell us about side effects.
So now given to many tens of thousands of individuals, maybe even 100,000 plus, these agents are very well tolerated. There is a concern in particular for these agents for propensity for allergic reaction or anaphylaxis has been seen in far less than 1% of patients. But there is a still in the EUA is a requirement of a 1 hour observation period, which we're trying after infusion, trying to get shortened, because if these events are going to occur, one, they're rare, and two, they're almost certainly to happen during infusion or in the first 30 minutes. But right now it's still a 1 hour observation period. There's really not been much else reported in the trial data or in the surveillance data that I'm aware of that would be a concerning finding related to adverse events. Again, with any new drug, there's still needs to be monitored as it's given.
So I'm sure, so as we've seen a lot of heterogeneity across the world in terms of the approach to vaccinations and offering vaccines, there must be heterogeneity in terms of the use of monoclonals. Are there countries ahead of us in terms of the use of monoclonals, and what are they doing? Yeah,
so I think, one of the challenges is these agents are pretty expensive. And so it's really, wealthier nations, essentially North America and Western Europe that are using these. I think the UK and the US have the most use and uptake of the agents. I think in the context of struggling to get many countries adequate access to vaccination, monoclonal antibodies are sort of not at the forefront of the conversation. And we just learned, because we're trying to see how in the the supply chain issues that have occurred over the last couple of weeks, what it would take to purchase out of pocket, because we never had to purchase these drugs. The federal government has purchased millions of doses of these to distribute for free. If you try to buy the GSK agent, it's $2,100 per dose. And maybe the federal government negotiated a better rate, but that's what they're paying close for the Lilly and the Regeneron agent. So I think it's just, it's a little bit challenging financially for other places. There's certainly regional differences within the United States. And this gets to the point of emphasis. The 70% of the monoclonals in the US are used by 7 states, and they are all in the Southeast, and they all have relatively low vaccination rates. So there's some concern here as we're trying to promote the strategy of treatment, of avoiding vaccination and only relying on therapeutics, which we don't promote, but is definitely promoted out there in some areas of the country and by some individuals. And so it makes it challenging. Our message is move prevention as far upstream as you can. Get vaccinated, and then if you need treatment, there is treatment available, but not wait until you get sick and then hope that treatment's available for you.
Good. Hillary, you have an announcement for an upcoming town hall.
And I'll call out Hillary has been an amazing partner in the MAB Colorado project. It's done a tremendous amount of work and effort with the dissemination and implementation activities. So thanks, Hillary.
I appreciate that. It's certainly been a tremendous team to work with. One of the roles I have is to increase provider, but also community member understanding and awareness of the availability of treatment. And we will be holding a public-facing virtual town hall on October 12th. So Holly has messaged me. We will send out that flyer with the DOM newsletter, and certainly would encourage you to reach out to me if you'd like the flyer sooner. I can get it to you as early as Monday. we will be very interested in having individuals who may have a transplant, may otherwise have be immunocompromised, so that they can ask Adit and Matt Winia and others questions about living with COVID and best options, including monoclonal antibodies, because we know that this information is really hard to process and important. So we're excited to have that community event, I think October 12th in the evening.
Good, thanks, Hillary. And last question, any estimation on when AstraZeneca monoclonal antibody may be available for immunocompromised prophylaxis?
I think that, so their data is only available, I believe, by press release. I don't think it's come out by even on preprint yet, but that has not stopped emergency use authorizations from occurring. So my understanding is that they have filed or are imminently filing for EUA, which will still take minimum a month to 6 weeks to get through. So maybe closer to the end of this year, December timeframe, something like that. November, December. Can certainly provide, I'll try and pull up as people are talking, at least the information on that trial, but it's the Provent 3 trial. Go.
So Adit just put the website up for the link up for the Provent 3 trial.
And that was 5,200 participants.
Wow.
They don't show for prophylaxis. They don't go into great depth here. Because we're studying this agent in Active 3, we have collaborations with these various industry partners as part of the project. So, but what I can say is that the effect seems to last for 6 to 9 months. So separation between groups continues for an extended period of time.
Good. Well, Adit, this has been incredibly informative. Thanks very much for presenting, discussing, and importantly, thanks for all the work that you're doing in this area. Really important work.
Great, thank you. Thank you for having me. Feel free to reach out to me or the team if there's questions. I think things change weekly, as I was showing, so we're trying to provide the most up-to-date information, but
right,
it changes.
Yep. Okay, good. Well, thanks again, and thanks for participating in the discussion, everyone. Good night.